Signal transduction is the process by which an extracellular signaling molecule activates a membrane receptor that in turn alters intracellular molecules, creating a response. It involves transmembrane receptor, which is one of the forms and important part of the cascade. G-protein coupled receptor (GPCR). G-proteins, also known as guanine nucleotide binding proteins. There are three phases of signal transduction. First, reception. A protein at the cell surface detects chemical signals. Second is transduction. A change in protein stimulates other changes, including signal transduction pathways. Last is response that involves almost any cellular activity.
As a receptor site for a small molecule called ligand exposed on the outside of the cell, the portion of the receptor on the inside of the cell can bind to the G protein that has guanosine diphosphate attached to its alpha subunit. When ligand bind to the receptor site on the outside of the cell membrane, the G protein changes conformation. And guanosine diphosphate replaces the guanosine triphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunit. This step can be repeated as long as the ligand remains bound to the receptor. When the ligand separates from receptor site, additional G protein no longer activated. Inactivation of alpha subunit occurs when its own phosphorylase activity removes a phosphate. From guanosine triphosphate, leaving guanosine diphosphate bound to the subunit. The G protein subunit then recombine and attach to the receptor in the cell membrane.
What happens if the signaling of G protein is deficient? The deficiency of G protein signaling can happen either by reduce the level of G protein or through the decreasing of signal initiation. When a decrease in the production of G protein happens, a disease such as night blindness will be triggered where the mutation in GT protein alpha subunits affect the response of rod cell to light. Besides, pseudo-hypoparathyroidism may be one of the effects of decreases in the production of G protein, where the genetic loss of GS protein alpha subunit result in the responsiveness to parathyroid hormone. Other abnormalities involve decreased signal initiation due to the inability of G protein to switch to active states. For example, the symptom of whooping cough pertussis result from the action of a bacterial toxin which adds ADP ribose to the receptor binding C-terminal of GI protein alpha subunits, causing a reduced responsiveness of G protein to receptor activation.
When excessive G protein arises. Excessive G protein signal can arise through either increased signal initiation or defective signal termination. Increased signal initiation occurs in dextrotoxicosis, where a mutation in the receptor for luteinizing hormone can overstimulate G proteins, resulting in excessive production of testosterone. In addition, Several cases of essential hypertension arise from mutations in G protein's beta subunits. Diseases arising from defective signal termination results from the persistent elevated activity of downstream effectors such as cholera, the symptoms of which results from the action of a bacterial toxin that adds ADP ribose to a G protein's alpha subunits to prolong the activation, resulting in the stimulation of adenyl cyclase and the subsequent excretion of salts and water, leading to fatal diarrhea. The other two diseases involved are adenomyomonas and also McCann-Albright syndrome.